Seldon Kwafo, and this summer I had the pleasure of working in the Hua Lab under the mentorship of Lei Young, and I'll be presenting my talk on the non-canonical function of structural maintenance chromosomes 5 in plant innate immunity. So plants are invaded by an array of pathogens, and thus they deploy mechanisms against these invasions. So understanding plant disease resistance is crucial to increasing our knowledge on plant immunity. In this presentation, I'll be talking about plant innate immunity, structural maintenance chromosomes, and my project as a whole. So plants, unlike animals, do not have mobile defenders, thus they rely on the innate immunity. So the first part of plant innate immunity, which is divided into two categories, involves the use of pattern recognition receptors to recognize patterns associated to microbes, and this recognition event has signaling cascades such as the production of reactive oxygen species and cell wall fortification. A second line of defense in a situation where the first line of defense is inhibited, you have the use of nucleotide binding leucine-rich repeats, which recognize pathogen effectors and can deploy a mechanism to boost the PTI immune responses, and also the recruitment of phytoalexin deficient 4, which promotes salicylic acid signaling. So to preface my project, I'll talk to you a little bit about structural maintenance chromosomes and what they do. So structural maintenance chromosomes complexes are key regulators of chromosome dynamics, structure, and function as well in DNA damage and genomic stability. So in the SMC5 and 6 complex, which is part of structural maintenance chromosomes, you have it specialized in genomic stability, and also it promotes DNA damage repair mechanisms. So in my project, I'll be looking at the SMC5 and the SMC5 complex. Studies in Arabidopsis have indicated that besides the canonical function of SMC5 in DNA damage repair, it is also involved in regulating of plant development and abiotic stress responses. But the role of SMC5 in plant innate immunity has not been studied, since the SMC5 mutants are embryo lethal and do not produce viable seeds in order for us to study this more. So through genetic screening, my lab has been able to identify SMC5 plays a role in plant immunity by using a weak allele. The tDNA knockout insertion line of the SMC5 is embryo lethal, so our lab has the allele that we can actually study SMC5 and its role in plant innate immunity. So in order to further explore SMC5 and plant innate immunity, we wanted to look at how to characterize the SMC5 mutant. We noticed that the SMC5 has pleiotropic phenotypes. We observed that the SMC5 mutant has a short shoot phenotype, and as well as we did a quantification and looking at the fresh weight, and you can see here that there's a decrease in weight compared to the wild type Columbia. And also we had a complementation line, which is to verify that the SMC5 mutant indeed has this phenotype. The complemented lines were complemented with the wild type SMC5, which reverted into the wild type phenotype, as you can see here, compared to the wild type. Another growth phenotype that we observed were the root morphology, and as you can see here, there is a shortening of the roots compared to the wild type, and as I did a measurement of the root, and clearly you can see there is a difference between the root length and with the wild type and SMC5. So to observe the immune responses in the SMC5 mutant, we did a pathogen assay with Pseudomonas syringae, where after 3 days post-infection, we saw in the SMC5 mutant that there was an increase in disease resistance compared to the wild type. The SMC5 mutant had decreased susceptibility in reference to the wild type, as you can see clearly here. And as I mentioned earlier, hydrogen peroxide is produced during different pathways, and in order to detect hydrogen peroxide, we use diaminobenzadiene to detect the presence of hydrogen peroxide in the leaves. So as you can see here, the SMC5 mutant has a higher accumulation of hydrogen peroxide compared to the wild type, and this shows that there is a defense response without pathogen infection. So this may also contribute to the growth phenotype we saw earlier on. So it's commonly observed that plant growth is inhibited by elevated defense responses. We saw the characteristics where SMC5 has a growth defect such as dry shoots and short roots, and there is an increased disease resistance in the SMC5 mutant, and we saw that there is higher accumulation of hydrogen peroxide in the SMC5 mutant, and this may be due to elevated defense responses without pathogen infection. So these characterizations led us to the next hypothesis. Does PAD4 function contribute to the SMC5 mutant phenotype, where PAD4 contributes its a mediator that is in salicylic acid signaling in defense responses. So we observed that in the PAD4 double mutant compared to SMC5, there was a growth difference where PAD4 was able to suppress the SMC5 mutant phenotype and partially rescue the SMC5 phenotype. This means that the SMC5 mutant largely depends on the regulation of PAD4. We also had a growth weight quantification where we can see a huge difference between SMC5 mutant and the PAD4 double mutant, which correlates to the phenotype we see here. After 3 days post-infection, we observed that the double mutant had bacterial susceptibility compared to the SMC5 mutant, and this correlated to the growth phenotype that I showed earlier on. So the PAD4 mutant also had a low accumulation of hydrogen peroxide, and since PAD4 partially suppresses the SMC5 mutant phenotype, the accumulation indicates that the growth and immune responses is largely dependent on PAD4. So now we know that PAD4 contributes to the SMC5 immune phenotype. As you can see here, that the PAD4 partially rescues the growth phenotype, as I showed you earlier on, and the double mutant has a low accumulation of hydrogen peroxide, similar to the wild type phenotype, and the growth and immune responses is largely dependent on PAD4. So aside the fine-tuned immune system, plant immune system can be lopsided by misregulated NLR genes, which I mentioned before, that recognize pathogen effectors. So in order to categorize SMC5 as an autoimmune mutant, we wanted to find out that if the NLRs that are activated in the SMC5 mutant leads to the phenotype that we see. So in this study, they did, so the proposition is that autoimmunity may result from upregulation of NLR genes. So we adopted a strategy to identify the NLRs in the SMC5 mutant, which was done by this study where the Capt3 is an autoimmune mutant, and by putting in a dominant negative form of the NLR genes, it inhibits the wild type NLRs in the mutant and restores the wild type phenotype. So we decided to adopt this approach in order to find the NLRs that are in our SMC5 mutant. So to explore this hypothesis, to To explore this hypothesis, we used 108 Arabidopsis dominant negative NLR genes, and we had the plants growing for about 4 weeks, and then we prepared the strains after the plants were were bolting to be used for transformation, and we did a transformation through floral dipping and to generate transgenic plants. So now we are currently growing the plants to obtain the seeds. Then once we obtain the seeds, we are going to screen for the plants on the selection plate to identify which NLR gene is responsible for the phenotype we see in the SMC5 mutant. So autoimmune mutants with constitutive defense responses often have reduced plant growth, as we saw in the SMC5 mutant, and compared to our wild type. So as we can see here, SMC5 mutant has a growth and immune phenotype, and we notice that the SMC5 mutant has pleiotropic phenotypes that are largely dependent on PAD4, which is a downstream mediator of immune responses. And through screening, we will be able to identify which NLRs are responsible for the growth and immune phenotype seen in the SMC5 mutant, and observing protein localization will bring us closer to understanding the role of SMC5 in plant innate immunity. Thank you. I would like to thank my PI and my mentor for giving me this opportunity to be in the lab and helping me achieve independence in this project that was designed for me, and as well as all the members of the lab, Delaney and George, and NSF and USDA for funding this. Thank you.
Wait, we have questions?
Questions?
I have a question. So you said that your hypothesis is that because the stress response pathway goes into overdrive in the SMC5 mutant, that's why it's smaller. The plants are smaller. But do you think it could be the other way around as well? It's like the plants are smaller for some reason, and that puts
we are trying to observe. So we've seen these characteristics that are expressed in the SMC5 mutant, and now we're trying to find out what's responsible for that phenotype. So we know that upregulation of NLR genes, that resistance genes, can contribute to the phenotype that we see. So we're trying to find out if mutant is an autoimmune mutant and it produces those phenotypes based on the upregulation of those genes. Any questions?
You had a question here?
So I saw in your methods that it's probably my fault, you flew by through the methods of like floral dipping.
Yes. So floral dipping is where you have the female dominant of Arabidopsis. It's a method of transformation where you dip the plants in resubstantiated agrobacteria, and with this method we are able to produce transgenic plants through the transformation by dipping it into a resubstantiated agrobacteria. Any questions?